Well, good afternoon, or for some of us, good morning to this session, the impact of pesticides on ecosystem and human health risk assessment based on data from the Sprint large-scale monitoring campaign. We have a very tight schedule today, so I would like to start with our first speaker, that is Violette Geissen. She is the project coordinator of Sprint, and I will put on her presentation now.
Yeah, hello everybody. We already had a Sprint session last year at the UN Science Summit. This year we will give you further results of the very important issue about pesticides and the risk of human and environmental health and how we address this. This year I will show you the Sprint approach for global health risk assessment. The aim of our Sprint project is to develop and test an integrated global health approach to assess the risks and impacts of pesticides on the ecosystem, plant, animal, and human health. And we want to identify transition pathways towards sustainable use of pesticides. If you are interested in more, you can look at the video, so then you can see what we are meant to do. The Sprint project is already running for 4 years, so we have very important output already. From the first period, we have the outcomes of a large-scale monitoring across Europe of pesticide residues in ecosystem and human matrices, and we did it as well for Argentina. Furthermore, we have characterized in the first year the residues. In this session this year, we will show you how we try to assess the exposure and risk to ecosystem and humans derived from real-world data. A little bit coming back to the field campaign, we conducted a field campaign in 2021 in 10 case study sites all across Europe and in Argentina. We took a huge amount of samples from the ecosystems. That means water, sediment, soil, air, and indoor dust. From humans, we had more than 670 participants, and we tested more than 200 different pesticide residues, from which 32 were approved for dual use. And we had here one synergist. Take it into consideration. Here you can see the huge amount of samples we took in total from environment, humans, and you can see here that we took in total 1,800 environmental samples, 1,400 livestock, and more than 4,000 human samples. For human, we took blood, urine, and feces. And here for the environment, you can see all the samples we took. If you have more questions, you can address them and send us always an email to get more information or look at our papers. Next one. What are the main outcomes of this unique sampling campaign conducted in the world is that residue mixtures were identified in 100% of water, air, and indoor samples. So there were not only one sample which only contained one pesticide. 96% of the soils from conventional farming as well contained mixtures, and even 79% of soils from organic farms. What about crops? 85% of conventional crops contained mixtures and 40% of organic crop samples. If you want to see here the numbers, they are presented here. And what obvious here is, if you compare soil, crop, air, indoor dust, and water samples, we saw that in organic farms, here mentioned as an O, we already had less samples. We always had less numbers of pesticide, and that they strongly accumulate in indoor dust, showing here for conventional farms about 50 to 100 different pesticide residues in one house. That means that it's all related to the presence of samples being present, residues being present in one household. Next. For urine, I will only really give you one example here, taken from my colleague Hans Mohl. You can see as well that in urine samples, mixtures of residues, here you can see the numbers, the maximum number we detected was 16 residues in one urine sample. Residues were omnipresent in urine samples of humans. Next one. So, we analyzed as well feces, and we could show that that urine and feces are internally showing the internal exposure of humans to a wide range of pesticides, not only through food residues, but also from other sources and routes. Exposures to mixtures is a rule rather than an exception. What do we do with this data now? Here's a sprint approach to assess the environmental risk. We, first of all, did a prioritization of mixtures to be tested in a tiered approach. You can imagine that it is impossible to test all mixtures which we have detected to be present in the environment. So we had to follow a prioritization approach, which Kayode will present us. And what we did, we compared mixture effects with single effects reported by EFSA in aquatic and terrestrial ecosystems. So what would EFSA say? Would these concentrations of pesticides we detected in soils and water would cause a risk for aquatic or terrestrial life? And how is this compared to the real situation in field? For the human exposure, we were looking to non-dietary exposure, taking wristbands and active air samplers into consideration, and we conducted a dietary exposure DPA to explain or to try to explain where the pesticides come from, if not from food. Further on, we tested the health risk, the human health risk, based on in vitro and in vivo tests with new indicators. My colleagues now will show you these approaches and the first results we have based on these exposure and risk assessments. So please let you surprise by the presentations of my colleagues. And please, I'm free to answer your questions. If you want to give your question in chat, please let me know what your questions were. There's still, I think, some time to answer your questions. If there are no questions in chat?
At the moment, there are no questions in the chat. So I think if there are no questions, then we just continue to our next speaker. That will be Kayode. So we just saw the omnipresence of pesticides, and now we dig deeper into what is this effect of these pesticides. And well, Kayode will be the first one to show this in earthworms. So Kayode, the floor is yours.
Oh, thank you, Paula, for the introduction, and thank you, Violette, for the general introduction to this topic. So I'll be talking on the cumulative mixture risk of pesticides to native earthworms. As part of the sprint, as one of the main focus of the sprint project, we're trying to look at other species other than standard species to see the effect of pesticides on them, and then to see if we can use if regulators or risk assessors can actually use those kinds of data to protect the soil or the environment. So, okay. Okay. So soil functions are impaired. As we can see, there are threats to terrestrial ecosystem. And one of these ways are through pollution of the soil, especially by pesticides. So pesticides has been very useful in the farm, but as well notorious for being, toxic to non-targets in the environment. So, and because they are designed to be harmful to organisms, especially to target, the non-targets are usually caught in the crossfire, which includes, mesofauna in the soil, microfauna, pollinators. Many of these pollinators have their life cycle, part of their life cycle in soil, so they are also affected. Okay, So for the sprint, we're looking at pesticide risk assessment. And as we know, we presented about this last year as well, that risk assessment is not very realistic if we just depend on single pesticide and data. But then for sprint, we are looking at mixtures of pesticides, because in the real sense of it, just like Philipp also introduced, pesticides are the rule, not the exception. Pesticide mixtures, sorry, are the rule, not the exception. So this is the approach for sprint. So looking at, mixtures of pesticides and their effect on terrestrial ecosystem. So in our case, we are using the earthworm, right? So for sprint, we have case study sites, which is 10 European countries and one non-EU country, which is Argentina. But then for this particular study, we're looking at 5 different case study sites representing different cropping systems, as you can see here on this map. So quickly, because of our time, I will go on. So these are the case study sites. So they are represented by country. So it's not just for people to be aware. It's not that if you see that one country is toxic, doesn't mean that it's really toxic. Based on our case study sites, based on how we chose the experiment. So that's what we have here. All right. So the methodology. So for this particular test, we try to replicate, or we try to do the same thing that happens in the environment. So farmers spray pesticides, sequentially, there's a spray schedule over the season. So we try to do that in the lab as well. So we've sprayed, we expose our organisms in the soil, and then we sprayed with pesticides following, recommended application rates over time based on the spray schedule. And that's exactly what we did. So on the left of my screen, so the smaller earthworm here is a native species, which is Lumbricus rubellus. It's an epigeic earthworm species. That is, it usually feeds or stays on the top of the soil. While here on my right, based on my screen, the bigger worm is the Lumbricus terrestris, which is an anesic worms. So their niche is always very deep in the ground, and then they come to the surface to feed. So these 2 species, and they are all European native species. So there are data for these species are usually very scarce. So these are some of the new sprint species that we're trying to look at the effect of pesticides on them. So very quickly, because of time, I go to the results. So after exposing these organisms to pesticides over time, so we can see our results here. Survival of this species was impacted, as you can see with the asterisks on the graph on the Netherlands case study site. And then for the reproduction was impaired in all of the sites. We only found very little reproduction in the Czech case study site, but for the other ones, they were all 100% inhibited, which means that there was an effect of pesticides on this species. The control was the only one that showed that there was reproduction. Okay, so that's on rubellus. On terrestris, the Portugal site was impaired for the survival and the cast, and as well as the cast. But then growth was impaired from all of the case study sites except Spain. So growth was the most sensitive endpoint here. So this is the pesticide residue accumulation over spraying schedule. So this is like a model of what will happen based on this on the cumulative pesticides. So this is concentration over time. As you can see, Netherlands and Portugal, they have the highest cumulative. That's about 0.035 and above, as you can see, and 0.003 for Netherlands. But the others, they have lesser compared to Netherlands and Portugal. Maybe that's the reason we're seeing toxicity most in both of these case study sites compared to the others. And then one other thing, you can see the number of active substances in Netherlands and in Portugal are they are higher than others, about 10 different active substances and 10. So maybe that's the reason why we are having higher accumulation and also high toxic effect. So discussion very quickly. the case study sites with more pesticides or more active, substance applied had more impact on the earthworms. As we can see, the Netherlands 10 pesticides and Portugal 10 pesticides as well. So cumulative pesticide residue levels were highest for Netherlands and Portugal, and they were lowest for Spain. So hence, earthworm in Spain, it had the least impact, as you can see. The one in red, which did not the growth of, for example, terrestrials was not affected at all in Spain, as as I showed previously. So, and then what we saw with this studies also, earthworms were impacted differently by the pesticides. So, for example, the case study sites of the Netherlands impacted the Hell rubellus, the epigeic earthworm, more compared, and then but in the case of the L terrestris, which is the anesic worm, it was the Portugal case study sites that impacted it more. So there could be some reasons for that, but that's just to show that they were impacted differently by the pesticides. Growth as an endpoint was very sensitive for terrestris, but not for rubellus. So these are some differences in these species. So conclusion and way forward. So we have shown that pesticides can be, toxic and can cause serious problems to terrestrial biodiversity, even at recommended application rates. So this experiment was based on recommended application rates applied over time, following normal spray schedule. So, and you can see, and we'll be able to demonstrate that. Then current pesticide risk assessment method should incorporate mixtures effect of pesticides. This is the essence of the Sprint project. And as we can see there, we can see, for example, you could see that the pesticides, the case study sites with more pesticides, more active substance, were more toxic, and which led to, cumulative effects. Native species representing different niches should be added to the existing suite of standard test species for generating data for efficient risk assessment of pesticides. So this is what we've been able to show, and then we hope regulators will take this into consideration. Thank you, and that's my presentation. If you have any questions, you can throw it in the chat. Thank you very much.
Oh, there's a question coming. What kind of pesticides you examined? Conventional pesticides or also biological pesticides?
These are conventional pesticides.
Yes, and that's because Sprint targeted conventional pesticides, so the synthetic pesticides and not the biological ones, which is, of course, also still very interesting. So a lot more work to do still, I would say. Well, thank you, Kayode. I think because of time, we go on to the next speaker. So we go from soil to water. And I would like to give the floor to Nelson Abrantes from the University of Aveiro in Portugal. The floor is yours.
Thank you, Paula, for the introduction. Hello, good morning. I'm Nelson from the University of Aveiro, Portugal. And following the previous presentation, I will address the topic assessing the impacts of realistic pesticide mixtures on aquatic ecosystems. So, as you know, pesticides, they are designed to protect our crops, but they frequently reach water bodies, namely through runoff, leaching, spray drift, and wind drift. Once the pesticides enter into the aquatic systems, they disrupt the balance of the ecosystems, affecting a wide spectrum of species, from primary producers to top predators. A recent data from the European Environmental Agency highlight that 22% of European surface water sites exceed pesticide thresholds, underscoring the urgency to address this topic. As you may know, current agricultural practices, they comprise multiple pesticide applications. Farmers apply formulations with several pesticides, and they often undergo multiple applications along the year. And this results in complex mixtures of pesticides in the environment. A monitoring study conducted also by the European Environmental Agency, over 10,000 aquatic ecosystems reveal the presence of multi-pesticides, ranging from 10 to more than 100 pesticides in a single water body. As presented by my colleagues, the sampling study conducted in sprints, where we analyzed 64 aquatic systems across 10 European countries and Argentina, also revealed the presence of several pesticides residues in surface water, from 8 to 40 compounds in one single system. Active substances, new pesticides, can only be approved for use if they meet specific criteria, being tested for its ecotoxicity. However, this is done just for single compounds. Considering the presence of complex mixtures in the real world, we are potentially underestimating the risk of pesticide mixtures for the aquatic ecosystems. And this situation raises particular concern based on the assumption that exposure to multiple pesticides poses a higher risk as distinct compounds can interact, leading to synergistic effects. So the question that we raised in Sprint was, how do freshwater non-target species respond to exposure to multiple pesticides? And for that, we defined the following testing strategy. Based on the pesticides we found in aquatic systems, in each 11 case study of Project Sprint, we define a mixtures consisting of 5 pesticides that were the most hazardous to aquatic species and the more frequent. Here we have all the distinct mixtures we tested, consisting mainly in herbicides and fungicides, but also some insecticides, all them synthetic. Each mixture was tested at 4 different concentrations plus a control with no pesticides. We include the measured environmental concentration, that is a realistic concentration. Also, the PEC, the predicted environmental concentration calculated by the focus model, and also 3 and 5 times the PEC concentration to establish a dose effects response. In our study, 5 aquatic species were tested, including organisms from different trophic and functional levels. So the species that we tested, very briefly, 2 producers, a microalgae and a macrophyta, where we look at growth inhibition, and we also tested on animals, a pelagic invertebrate, Daphne magna, a benthic invertebrate, and also on zebrafish. For the animals, we look at the survival when exposed to the 11 mixtures. So since this results in so many experiments, I will show you a summary of the main findings. So for the microalgae, we observed that more than 50% of effect in the predicted environmental concentration in PEC was observed for Czech Republic, Slovenia, Croatia, and Portugal. In the case of macrophyta, we saw more than 25% effect when exposed to the PEC for Netherlands, Czech Republic, Sweden, and Portugal. The Daphnia species registered more than 50% of effect in the PEC concentration for Slovenia, France, and Spain. The benthic Chironomus show more than 40% effect in the measured environmental concentration for Argentina and Portugal. And let me remember you that the MEC is the concentration that we measure in the fields. And finally, the zebrafish was less affected species, showing a maximum of 25% effect when exposed to 5 times PEC in the case of France and Spain, and no effects on survival was observed when exposed to the other case studies. So all the tested species were affected by pesticide mixtures, but in a different way. And important to say that all these assays that I show you were performed using species and endpoints defined by EFSA for the authorization of pesticides. However, in the Sprint project, we also aim to introduce new endpoints and evaluate the response at sub-individual level. So for the zebrafish, that was the less affected species in terms of survival, we decided to go further and we assess the neurotoxic effects as well as the behavior responses when exposed to the 11 mixtures. Okay, so in the case of neurotoxicity, we measure the acetylcholinesterase activity, and we also assess the changes in behavior. And for that, we look at motor activity and swimming patterns. Okay, so looking now at the results of the neurotoxicity, we observed that cholinesterase activity significantly decreased under all tested concentrations for most of the case studies. And this decrease in cholinesterase activity is particularly important because it is related to hyperactivity in the tested species. Okay, regarding the zebrafish behavior, namely the locomotor activity assessed by the swimming distance, we observed that there was an impact on locomotor activity for the case studies of Portugal, Croatia, and Czech Republic, with all of them showing increasing swimming distances. These cases also correspond to a significant effect on cholinesterase activity, reinforcing the connection between neurotoxicity and changes in behavior. And finally, regarding the swimming pattern, we observe a tendency for the larvae to spend more time in the outer zone under unfavorable conditions. And this was noted in multiple countries and is correlated with a decrease in cholinesterase activity and increase in swimming distance. So moving now into the conclusions of this study, all tested species were impaired when exposed to the 11 distinct mixtures. Significant effects observed at the predicted environmental concentration highlight the potential hazard posed by pesticide mixtures in aquatic systems. The observed toxicity was dependent on the species tested and on the formulation of the mixtures. And although the PPP mixtures had a lower impact on zebrafish survival, they significantly affect the cholinesterase activity, indicating a neurotoxic effect that leads to behavioral changes. So let me leave you two main take-home messages. Sub-individual parameters can be more sensitive to pesticide exposure than individual measures, and these parameters can act as early warning indicators for potential toxicity. So incorporating sub-individual parameters and points into environmental risk assessments, incorporating them, we can detect risks earlier and achieve a more accurate evaluation. And the second take-home message, our findings highlight the urgent needs for a more comprehensive investigation on the impacts of real mixtures on aquatic systems. And this is critical for safeguarding clean water sources and ensuring universal access to safe water, contributing to achieve the Sustainable Development Goal 6. And also, the observed effects on aquatic species underscore the importance of protecting freshwater life and ecosystems and reduce pollution in aquatic systems. So we are also contributing to achieve the Sustainable Development Goal 14, life below water. So many thanks for your attention.
Thank you, Nelson, for your presentation. There are some questions in the chat. I would like to start with a very interesting one that is asking, what do you think is the role being played by the soil?
The role playing by the soil is for me or for Kayode, but I can answer.
It's for you. I think it's for you.
All compartments are connected, and so what is on the water comes usually comes from soil, also air, but mostly from soil. So aquatic pollution is completely dependent on the agricultural practices. What we can do is, of course, and is a contribution of Sprint, is to show that we have real impacts on all compartments and the need for go to more sustainable agricultural practices.
I would
you,
like to add
Violette.
here that there is as well a very important issue that the actual models that predict the transport from the soil to the water bodies are mainly based or mainly taking a transport of only soluble pesticides into consideration. So following the models, the main transport is based, if it's raining, runoff water is entering into the water bodies. But the pesticides which are attached to soil particles, they are transported with water erosion, and that means attached to the soils. And this part as well enters into the water bodies, being then accumulating in the sediment of the water bodies and being there of harm for many, many years. So this part is not sufficiently included into the actual regulation for pesticides.
Yeah, and one of the questions is a remedial solution, in my opinion. And since soil is so different, the solution is really to go for more sustainable practices.
Okay. So I would like to remind the participants that they also can speak, but questions via the chat is also perfectly fine. Well, thanks again. Then we move on to soil again. We go to Philipp. He's from the University of Hohenheim in Germany. So take it away.
Thank you for the introduction. So we're going back to soil, and this time we will look at the effects of multiple pesticides on soil microorganisms. Why is this important? As we also just discussed, soil microorganisms are among the first non-target organisms that get in touch with the pesticides, and they also serve as major degrader of those. Additionally, they provide very important ecosystem functions, such as this degradation of pesticides, but also for nutrient cycling. The official tests on soil microorganisms are limited and outdated. There are only 2 tests for this. So our aim was to test new indicators that include those special microbial functions and to improve the mechanistic understanding what lies behind the effects on soil microorganisms. So what did we do? We spiked soil with pesticides and incubated them for 56 days in microcosms that you can see in this picture here. And we determined the microbial biomass, the community structure, so is it fungi or bacteria, as well as determining the activity by respiration and by the activity of enzymes that are linked to nutrient cycling. Also, we determined functional genes that are involved in pesticide degradation. So for this, we also used indices, which I just quickly will explain. We used the metabolic quotient, which is the respiration carbon to the biomass carbon, and this is used. So if this quotient increases, it is associated with a stress reaction of the microbial community. The carbon use efficiency is an indice that is used to see if the microbial community has a higher energy demand of a given substrate, or that it can utilize this substrate for growth. So if this indice decreases, we have a higher energy demand and less growth. And lastly, for the specific enzyme activity, this is just the enzyme activity divided by the microbial biomass. With this increasing, it indicates a higher nutrient demand depending on which enzyme we looked at. In the first experiment, we tested what MCPA and difenoconazole do on the degradation of glyphosate and how it affects the soil microorganisms. For this, we used 13C labeled glyphosate, as well as a herbicide called MCPA and a fungicide called difenoconazole. We all know glyphosate, and MCPA is a model substance for xenobiotics. It is very well studied and fairly easily degraded. Difenoconazole, on the other hand, is a fungicide that is more persistent. Next to control without any pesticides, we use 2 glyphosate treatments. This is 20 milligram per kilogram and 10 milligram per kilogram. This is roughly 0.75 of the recommended field dose, or 1.5%. This bottom glyphosate treatment with 10 milligram per kilogram, we then mixed with either MCPA or difenoconazole to get 20 milligram per kilogram of total pesticide load. And then we had a triple treatment with glyphosate, MCPA, and difenoconazole, also having 20 milligram per kilogram in total. So the effects on the microbial activity were as follows. Especially MCPA increased the metabolic quotient by about 35% in the first week. This is in line with the reduction of MCPA residues, and they declined towards day 14, and then it was below the limit of quantification. So this means that MCPA can induce a stress reaction in the first week or when it was present. Another problem is that the tests are often too short. You can see here day 28 and day 56. While most tests are only going until day 28, we would not see an effect of some of the effects we would not see if we would terminate the experiment after 28 days. However, we can also see an effect after 56 days. Another interesting finding was that the presence of MCPA increased the degradation of glyphosate by up to 40% in the mixtures. On the right, you can see a possible explanation, which is that if MCPA is either entering the soil, you see that on the left picture, or it is already in the soil on mineral surfaces, it can increase the bioavailability of glyphosate by either blocking the glyphosate of binding to mineral surfaces or by exchanging it. So there's an interaction between those pesticides. We also found that the carbon use efficiency decreases in the mixtures on various days. So there is a higher energy demand of the microbes while limiting the growth of those microbes. In a second experiment, we then looked at the effects of 2 other pesticides named prosulfocarb and boscalid on the substrate utilization of maize, where we had 2 different maize treatments. With one was fresh maize and the other one was aged by washing it with water. So we used prosulfocarb as the mode of action inhibits lipid synthesis in plants, and we thought this might also be the case for the microorganisms, and a fungicide, boscalid, which is frequently found in the case study sites as well. So we had the single treatments with the recommended field dose of boscalid or prosulfocarb, as well as the mixture of both. And as I just stated, the intentional idea was correct. Prosulfocarb actually inhibits the new formation of membrane components in bacteria, thereby also reducing the growth of the bacteria in the soil. Further than that, it also increased the specific enzyme activity involved in carbon and phosphor cycling, corresponding also at day 7. This means that the nutrient acquisition strategy of the microbes changed because they need more carbon and phosphorus. So to conclude, pesticide mixtures can influence soil microorganisms in either synergistic or also antagonistic ways. And more importantly, pesticides can affect those specific functions of microorganisms, such as glyphosate degradation. Next to the number of the pesticides present, also their mode of action can influence the microbial community and have an effect on non-target organisms. Also, 28 days of testing is often too short, so we'd recommend to extend the duration of the tests to have all the effects. Thank you for your attention.
Thank you very much, Philip. There's one question now in the chat. How can we reduce their effects in the soil?
Yeah, maybe we can, yeah, I could answer maybe here. I think what we found out is that in all soils we have analyzed, or nearly all soils, we have mixtures of pesticide. But there we have the very, very old ones, the DDTs, which were forbidden in Europe since 50 years. They are still there. And surely we have the new ones. So it's very difficult to get rid of the old ones because there is no remediation technology that remediates hectares and hectares and thousands of hectares of soils which contain DDTs, HCBs, or the dirty dosant. But what we can surely do, we can reduce the pesticide applications which we apply now.
Thank you, Violette. Then there's a last question. It came in via another channel. It's, is it not more novel to consider formulation of more biopesticides to reduce bioaccumulation effect of synthetic ones in humans during residual effects from agricultural produce? Dr. Peter Onua from Nigeria.
Surely it would have been very fantastic to as well analyze this, and we think that is the future as well. There are new calls related to these effects of biopesticides and the relation with the synthetic ones. In the sprint, we only considered the 200 synthetic ones, and this was an answer to the call text. I think it's very important to have as well a look on the biopesticides and especially find regulations for biopesticides which are not in place actually. But our project was about the effects of the synthetic pesticides. But I agree with you.
Thank you, Violette. Well, thanks again, Philip, for your nice presentation. According to the program, now Freya would have had her presentation. Unfortunately, due to personal circumstances, she was not able to join us today. So we skip this session and we go to the next session that is from Eoin Gunnigle. I hope I pronounce it correct, probably not, from the University of Cork. And he's going to tell something more about social behavior. So the floor is yours.
Thanks, man and Paula. Can you hear me and see the slides okay? Perfect.
We can see them.
Okay, great. So thank you for the opportunity to give my presentation today. So a little bit of a tangent from the soil and ecosystem health. So I'm just going to be presenting some of our data today from UCC in Cork on the impact of glyphosate in terms of altering the microbiome and the impacts on social behavior. So I guess just in terms of context of where we fit into the project and our relevance is differing to the ecotoxicological and toxicological tests. We're more involved in new sprint indicators for assessment effect on human health. So these are new models that could be interest or risk assessment, but are currently not yet embedded in standardized guidelines. So they emphasize multiple endpoints and they present potentially a more realistic scenario, or realistic scenarios being expected to result in solid or accepted evidence-based information for risk assessment. And in our research laboratory in Cork, we're primarily focused on the gut microbiota brain axis. So I guess just in terms of context and background, microbes have been an essential part of human life. We've co-evolved with microbes and established a mutualistic symbiosis. And there's accumulating evidence that has unveiled a bidirectional communication pathway between the gut microbiome and the central nervous system, referred to as the microbiota gut-brain axis. And so because of the interest and development of research, we've looked at developing therapeutics to meet the challenges of neurological diseases, and this requires an in-depth understanding of mechanisms by which microorganisms can influence the host nervous system, and so forth. So we're interested in these, and in addition to using, and testing our hypotheses on humans, on people, we also used standardized model systems like mammalian models using mice, and even earlier what Nelson mentioned with zebrafish, and even simpler systems such as Drosophila and C. elegans to understand the mechanisms by which microorganisms can impact and modulate host behavior. So there's a series of mechanisms or pathways by which the microbiota can interact with the host. These include the modulation of the immune system, the vagus nerve, the enteric nervous system, neuroendocrine system, and also production of actually neuroactive substances and metabolites by the microbes that can actually transfer across the blood-brain barrier and actually send signals to the brain. We're interested in Cork looking at the impact of the gut-brain axis throughout life. So it's evident that microbial colonization in the gut in early life enables the production of essential amino acids, vitamins, which happens obviously very early until the microbiome kind of reaches stability in adulthood. But these periods of critical windows and changes in the microbiome are obviously very important to us, and how we answer questions about how the microbiome is influencing our host behavior and health overall. We use these model systems such as murine models, and in terms of how our pathways that can change the gut and ultimately change host health and behavior, there's a variety of different factors, but this includes our environment. So it's a dynamic system shaped by exposure to different environmental factors. Obviously, diet is a huge one, but exposure to xenobiotics such as pesticides is also an important player. And there's a growing body of evidence and literature to suggest that the effects of pesticide exposure can manifest itself in changes in behavior as well, which we're very interested in our research center. And we recently published a paper on this, looking at pesticide exposure and the impacts it has on the microbiota gut-brain axis. There's a huge amount of information that exists and publication that kind of underlines what we know, that pesticides can have a negative impact on the gut microbiota. And there's a paper that I've highlighted down below with Daniele Mandrioli's group, which is part of the SPRINT consortia, that showed that microbiome profiling, even at low dose glyphosate and Roundup exposure, can be altered. So there's a large body of evidence to suggest gut microbiota changes with the exposure to pesticides. And also when we look at behavior, pesticide exposure has been shown to impact host behavior such as anxiety, memory, social interaction. The mechanisms underpinning these pesticide-associated behavioral alterations remain kind of inconclusive, and we're still trying to understand the mechanisms at play, and hence we're kind of very interested in bringing this research further. So a key question that arises, and one that we tried to answer in the project, is changes in the gut microbiota structure and function. Can these be driven by adverse effects, be driving adverse effects in behavior? So can they mediate the effects of behavior through this changed gut microbiome? And in this study, we focus at glyphosate. Obviously, it's been introduced before. I don't need to go into huge detail, but obviously the importance and prevalence of glyphosate in Europe, the sales are over a third for the herbicides in Europe, and the current levels of glyphosate in humans, in our environment, in the household, in soils and surface waters, it's extensive, and scientific evidence indicates that there's high concentrations of these pesticides found in even our groundwater. So this is a pesticide of very high concern, and one that we looked at very closely. So one model we used was an adult exposure model. So this is where we had a control group. So this was a group of mice that weren't treated with any glyphosate. And then we had a group of mice that were treated with the acceptable daily intake of glyphosate, which is 0.5 milligrams per kg per day. And this is defined as the daily intake of the substance, which over the entire life appears to be without adverse effects or harm to the health of the human. And we did this 10 times the ADI and also 100 times the ADI, which is around the no observed adverse effect limit. So In this study, we used a variety of behavioral tests. So I won't go into too much detail. We did an extensive array of behavioral tests, and we took obviously samples for different physiological endpoints as well. But just to focus on one impact, on one output that we found very interesting, was the 3-chamber social interaction test. So this is a validated test to assess social interaction between sex-matched conspecific mice, and allows for readouts for social novelty and social preference in mice. So obviously social interactions we know are very important and fundamental for biology. Social recognition is critical for structure and stability of our networks, and relationships that define social societies. So in this test, there are 3 phases. So the habituation phase, which is one at the top. There's a sociability phase, which is one in the middle, where we have a test mouse in the middle of the chamber, and they have 10 minutes to interact with a mouse that's the same as them in terms of it's the same age, same sex, or an inanimate object, which in this case was a rubber duck. And then the last phase of the test is a social novelty test, where you compare the mouse that was there in the second phase of the test and a new mouse, a novel mouse. And just to focus on the third phase, which was the critical outcome for this test, is if you can focus on just the white bars at the moment, these are the control mice. And what we would expect, each one of these dots is a mouse and how it behaved in terms of interaction time between the novel mouse and the mouse it was already familiar with. What you expect is that the mouse prefers interacting with the familiar mouse. Or interacting with the novel mouse, apologies. And you can see a significant difference here. However, with the mice that were treated with the ADI by 10 and also ADI by 100, there was a disruption of this novel social preference, which has been shown before in literature. It's not completely novel, but it's something that we found at a comparatively low dose that's been done in previous studies. And there's been a lot of research that has kind of talked or explained the impacts of the microbiome and social interaction. So what we found here was that glyphosate-driven effects on the cognition and general sociability below the NOAEL level. But what about the microbiome? What happened in these animals in the gut? Well, we found actually significant changes in gut composition. So this is just a bar chart. Each one of these colors represents a particular bacterial taxa, and each one of the total lines is one of the animals, the microbiome of the animal. So you can see, if you just focus on the green, the light green, these represent Lactobacilliaceae. So Lactobacillus, you probably are aware, are a very important probiotic species. So you can see a significant reduction of this group in the high dose, which is ADA by 100. And just to focus here, individual species, you can see a reduction in the dark blue compared to the gray, which was a control animal. So a significant reduction in beneficial bacteria. And you can see trends when you do things like this, which is a heat map, which tracks the most abundant species or taxa overall. And you can see differences between control samples and each level of glyphosate exposure, and even between glyphosate exposed animals. But in terms of mechanism, what we did was we took the fecal matter from mice that were exposed to glyphosate, and we transferred that microbiome into novel mice, into mice that hadn't had any exposure to glyphosate. And obviously we included a control group, which were given gavaged just water, non-glyphosate samples. So, and we also explored the behavior readouts in these animals as well, to see if the microbiome was a driver of these changes in behavior. And what we found was, if we're going back to the original result, that we actually did find the behavior phenotype in the microbiota transplanted mice. So this suggests, for one of the first times we've seen that glyphosate perturbed microbiome can also transfer alterations in social behavior in healthy individuals. So just to quickly summarize, glyphosate perturbed microbiome at the NOAEL can also transfer alterations in social behavior in healthy individuals. So this is a quite impactful result. We think it shows the potential of the microbiome mediated effects on behavior through pesticide exposure, but may have significant societal impacts on our overall understanding of how environmental exposure pesticides can potentially alter human behavior. So thank you for your time.
Thank you very much, Eoin, for your presentation. Are there any questions from the audience? I don't see anything at the moment in the chat. We are still on time. So hello, everyone. It's 4:05, at least in Europe, most of parts of Europe. So we would like to start with the second part of the sprint session. Another 6 presentations to go, and we start off with Peter Fantke, and he will tell us something about the global distribution of pesticide use. So Peter Fantke, please, the floor is yours.
Thanks, Paula. Thanks, Violette, for inviting me to contribute to your session today. My name is Peter Fantke, and I will introduce the global distribution of pesticide use as a data set between 2016 and 2020, with kind contribution from Dr. Eoin Gunnigle. So when we do a risk assessment like we do in the sprint approach, and when we do also environmental sustainability assessment as in many other tools, we actually need data on pesticide use. This is most important since modeling and also measurements to confirm what the impacts and risks are rely on the data what has effectively been applied in the actual fields. A global consistent data set on what pesticides have been used on what crops, at what crop stage, with what application method, however, is completely missing. In the FAO data set, we have pesticide use data. They are not linked to crop data. We don't know when they are applied, where they are applied, at what crop stage, how they are applied, and so on. And it does not cover anyway a level of detail that we need to understand individual pesticides risks on different environments and human health impacts. Now, what we present here is we looked at panel data globally. We complemented them with industry statistics and internal data from major pesticide producers like Bayer Crop Science. And then we curated them extensively to generate an input data set from more than 1 million data points across 5 years across the globe into a consistent data set of roughly 900,000 clean records for more than 120,000 combinations of pesticide, crop stage, and application method. Many of these data had data gaps that we had to fill. First of all, for several countries that are not so-called market relevant in terms of financial market, many African countries, for example, Iceland, Mongolia, that are largely also covered by deserts, for example, had no data at all. So we imputed them using the 900,000 clean data and tried to understand, looking at additional statistics like climate scenarios, like also what crops are grown where, what are the income from agricultural crop production, and so on. We imputed these data and filled the data gaps. In addition, where methods were missing, where crop growth stage was missing, we also imputed them using advanced machine learning method like XGBoost or conformal prediction. Finally, we combined all these data with spatial crop maps from 2020 to spatialize these national records of pesticide use, because we have also within a country like the United States or big countries, France, China, India, and so on, big deviations between the regions. Now, that is how it looks like. The starting point is a data set where we had in black records in the table that you see the reported data that are already curated, merging different data reported records together, giving also a confidence interval based on data quality and the variability within the data set for each combination of pesticide, crop, and so on. We have data on the dose and the mass applied in 1,000 kilos here for each application method, target compartment, crop stage, and so forth. And the red records are the imputed ones. So this is a very small excerpt of 120,000 composition records that we have put together here. The red one is the imputed ones that we predicted. Now, almost 3 million tons are applied as a 5-year robust average across the years 2016 to 2020. We included 180 countries, 130 crops, and more than 1,000 pesticides, which is all conventional chemical pesticides. We excluded the biological ones because they had incomplete records for most of the pesticides reported. China, US, and Brazil make almost half of the global pesticide use applied in terms of mass applied worldwide, although they only cover 1/4 of the crop area. So 54% are actually herbicides, Most of that, as you can imagine, is glyphosate, which makes a big share as an organophosphorus chemical. 70% are fungicides, 13 insecticides, and the rest are other pesticides like plant growth regulators. 10% are inorganic, mostly sulfur actually, and copper-based fungicides, and almost 10% are thiocarbamates, and the rest are again a multitude of many others. That is the granularity that the data set includes. So that is just one example where we have one chemical acephate applied in one country, US, and you see an inner circle distribution. What is the contribution of different crops that acephate in US has been applied as a 5-year robust average? Then within each crop, you see the crop growth stage, like 1019 is, for example, during leaf development for cotton, is 1/3 of the whole cotton data set is referring to that data set applied via a boom sprayer, mostly as the outer circle suggests, where we have for other crops, of course, also many other application methods. Most important is in the US, acephate has been applied mostly cotton and soybean. Other pesticides have a completely different profile, and that granularity is important to understand risk also. Now, when we put all this together, not just looking at one chemical, but more than 1,000 across the 130 crops we have, we can actually rank them in terms of pesticide mass applied. And when we look from the top right, the darkest purple bin is soybean glyphosate, actually. So it's the first row and the rightmost column. The small square is really the highest mass applied per hectare in one given country, for example, or across countries, actually. We have the same heat map per country across crops, and also for crop countries across chemicals. So what you see here is from the top right to the bottom left, that is a ranking in terms of what is the highest mass applied chemical. The top 3 here is glyphosate, sulfur, and mancozeb that you see here based on CAS numbers. And on the right side, you see soybean, maize, and the cumulatively other crops that are not specifically individualized here together make the highest share of the pesticide use globally. So on the right bars, the gray ones, you see the count of per chemical across many crops it has been applied. And glyphosate has been applied, for example, on more than 200 crops. On the top bars, you see the same thing. You see per crop how many pesticides are actually applied. And again, here there are several crops that really use more than 50, 60, 70 pesticides per year. Now, when we spatialize all this, we can see here another example. Atrazine, widely used, for example, in the United States and Brazil, actually banned in Europe. So you can see here also regulation and other aspects are influencing what we do. This is purely the reported data set. So the depth of the color shows the difference in the amount applied per hectare in terms of spatialized cell based on crop distribution maps that we applied here. Now, when we switch back and forth, you see this is reported data from aggregating and consolidating different data sets, including panel data, and that is imputed. So the chemical is very likely also applied in these countries, but is not included in the reported data set because these countries don't have a high market share globally. So they don't have any panel data or other, but the crops are grown there. So likely they are also considered here. So when we bring both together, we have actually per chemical and crop a global map of how the pesticide is distributed mass-wise spatially. And we have these types of maps for all 1,077 pesticides across 130 crops. Now, what we can do with that is, if we really want to have pesticide use combined with modeling, we see here on the x-axis from left to right, the pesticide mass applied. That comes from the global pesticide data, and we can zoom in per country and crop scenario, where we have on soybean in Brazil, 139 pesticides applied. From left to right, we see, on the right side, we have the high applied chemicals. On the y-axis, we need to then combine our pesticide use data with modeling as suggested earlier, where we have emissions and impacts, fate, exposure, and effects on ecosystems, for example. So on the bottom, we have low toxicity on the high and on the top, we have high toxicity chemicals. If we merge these together, pesticide mass applied per hectare times the impact per kilo applied, we get on the iso diagonal lines the total impact per hectare. So we need to see that not only the modeling, but also the pesticide use data quality is really crucial to arrive at such data. And of course, we can zoom out again and we arrive at our heat map again, just one block that we have checked here for the global pesticide use. I think it is important for prioritizing where we should reduce, which chemicals we should reduce, on what crops we should focus, and which countries should do what because of difference in the climate, economy, agricultural practices, and so on and so on. So the take-home messages are, we need consistent globally specialized pesticide use data because this is really key for meeting global risk reduction targets if we combine it with modeling. We need a high granularity that is currently really missing worldwide and full pesticide market coverage. 3 countries cover almost 50% of the global use currently, and we can combine the data with modeling to really arrive at the ranking from local to global scale. Thank you.
Thank you, Peter, for this nice overview. Really interesting. At the moment, there are no questions in the chat, but there are a lot of new participants. I would like to say, if you have a question, please raise your hand. You are able to speak up. So if there are no questions, then we continue to our next speaker. That will be Benjamin. He already put on his presentation. So, well, I would say, Benjamin, go ahead.
Okay, thank you very much. Thanks for the opportunity to talk here. So I will discuss a bit on the kidney studies we have been doing, and also a bit on the background behind that, and finally culminating towards an experiment which we conducted in the frame of Sprint, but for which we have results still in the process of being collected. So maybe as a short introduction to show the complexity of the kidney, here we see a kidney. It's responsible for a couple of functions in our body: general body homeostasis, that is one of its main functions, of course, waste removal, and also an endocrine function. It has a complex structure. It's built of over a million functional units, the nephron, which it consisted of. It's a tubular system, which consists of several segments, each lined by a particular tubular cell type, just to demonstrate how complex this organ is and how it is important to assess toxicity on this organ. This organ, the kidney, filters and concentrates urine. We know we filter about 100 liters of urine, of blood per day, but we urinate only 1.5 liters. So this is a highly concentrating organ. And by doing so, it encounters a lot of toxins, to which it's also highly sensitive. Now, when we're talking about kidney disease, what we are talking, how does it look like? This is the kidney here. This is a cross-section of the glomerulus, the filtering unit, and this is the sections of the tubules being cross-sectioned in this view. When we have acute kidney injury as possible, we have a lot of cell death. Cells really suffer from the toxicity, or we can also have development of chronic kidney disease, where the decay is more steadily. We have shriveling or shrinking of the tubules, and we get scar formation in between the tubules. This is best to compare like a scar on your skin. Now, knowing this, when we then jump back like 40 years into history, clinicians in Sri Lanka and El Salvador, Nicaragua discovered something they then termed chronic kidney disease of unknown etiology. In their wards, there were many patients suffering from CKD, but the cause was unclear. It turned out that these patients were mainly males, farmers living and working in agricultural environment, and there was no known cause for this disease. If we look at the same map of today, we see that more countries have reports, either by word or by proof, that this is happening and that there's this disease in kidneys is spreading in the farming communities. This is also recognized by the International Society of Nephrology, who also installed a CKDU network to investigate this. The cause and the mechanism is fairly unknown. There were two big theories at the time, either heat stress dehydration, because working in hot environment, or by the exposure of agrochemicals. Now, over the course of the years, the evidence towards a toxin involvement in this disease has grown by several epidemiological studies. And one of the more anecdotal stories is here, for example, in Sri Lanka, when there was traditional farming for years and years. since the 1980s, there was introduction of mechanization agrochemicals, and only like 5 or 10 years after, this disease started appearing, indicating that there was some kind of incubation period going on. So then we, from our side, we investigated renal biopsies from these patients, and we found something peculiar in the tubular system. We found these black granules due to particular staining, which we also observed on electron microscopy. So this is even smaller than a human hair. We're looking here inside the cells, and this turned out to be dysmorphic structures, which are normally not present in a healthy cell, but this turns out to be the lysosomes, or in regular wording, the stomachs of the cells. They seem to be constipated to a certain extent. The important findings we did was that the same phenotype was found in all places we investigated. So in El Salvador, even France, India, Sri Lanka, and Paraguay, we found such cases, and indicating that there's indeed the same condition going on in farming communities with highly prevalences of this lesion. The second finding, what was also quite important, was the fact that the lesion which we observed in these patients was the same as we observed in patients taking with a transplant kidney. Patients with transplant kidneys, they are taking immunosuppressive drugs to avoid losing their graft. And they take this kind of drug called calcineurin inhibitors. It is known for these drugs that they are nephrotoxic. So by finding this histopathological parallel between the unknown kidney disease and also in patients with transplant kidneys, we can say that the unknown kidney disease is likely caused by a toxin or a class of toxins. It also shows or allows to suggest that what we're looking at here, these dysmorphic lysosomes, can be used as a potential new indicator of nephrotoxicity. Therefore, we performed a rat study comparing dehydration in rats with exposure to a toxin, and we used the calcineurin inhibitor as a positive control. So it was a 4-week exposure study, either to cyclosporine or with dehydration. And what we saw was that in control and dehydrated, there was not much going on in the kidneys. However, after 4 weeks of cyclosporine, so the immunosuppressive nephrotoxic drug, we could already see the lysosomes appearing bigger in these tissues, and also seen the start of what is later to be found, chronic kidney disease. We also found on electron microscopy the same lesions as we found in humans, although to a certain extent they were larger and dysmorphic, but they did not show the clear mottled pattern they had in humans. So this made us to reason that, okay, there's a difference between humans and rats, obviously, but to what extent can we trust parameters in one species versus the other? Another big difference is that we performed a single exposure study. How about mixtures exposure study? So then we want to clarify this, and we went to literature, and then we realized there was a complete lack of real-world pesticide mixture studies. There are several studies performed with pesticide mixtures, but this is really not that common. And also we can question whether the sensitivity of current toxicity indicators is sufficient to really assess the risk. That's why in the frame of the SPRINT program, we designed a new study, pesticide mixture study in rats, but then based on regulatory as well as human exposure levels. So as a background, in SPRINT, as just was mentioned by the colleagues, several case study sites have been sampled. Therefore, we used from work package 2.3 the duplicate portion analysis. So part of the food, in part of the food, the pesticides present there were measured and identified. And this allowed us to calculate estimated exposures. Here we see the number of pesticides in the samples which were included, ranging from 2 up to 24 pesticides in the food of these households. We then calculated for each of these conditions the margin of exposure. Now, what do we understand by that? We have regular chemical testing, usually occurs at the NOAEL or the acceptable daily intake, so the safety levels. Then we have the real-world exposure. And then the margin of exposure is actually how far the real-world exposure is from the safety level. So it means that the smaller the margin of exposure, the higher the risk is of having adverse effects, or the closer to the margin of exposure. What we did is we calculated this for all the conditions here, and we identified the condition or the sample which had the highest risk, so the lowest margin of exposure. And it was this condition which we traced back to the original samples, for which you could identify 13 pesticides to be in it. And in these 13 pesticides were used in an animal study in several groups. We had 2 control groups to cover for normal and biological solvents. Then we have 1 group at the safety level, so the acceptable daily intake level, an additional group at acceptable daily intake divided by 25, an extra safety margin, and also 1 group included at the exposure dose, which we calculated directly from the food. So this is actually the human correlate of that experiment. We did urine and serum sampling over the course of 16 weeks. We included behavioral tests 1 week before killing of the animals. And then at the end point, we collected multiple organs and samples for collaborative assessment. So these analyses are now ongoing. In the urine, we have pH, molality, proteinuria, and several biomarkers. Also in the serum, we're looking for what might be there of adverse effects. There is many collaborative efforts going on to do heart, brain, liver, kidney, lung analysis. And also in the kidney, we're doing additional morphometric and molecular analysis to pick up more sensitive markers of adversity. So what the key messages mean from this side is that the kidney is an excretory organ, and it really sees a lot of chemicals, and it's also very vulnerable to them. There are kidney diseases out there which are linked to the use of pesticides. Causality has to be shown, but there is a high likeliness. This is part of the research. Then also translating health findings in rodents to humans remains challenging, but might be improved by setting up animal experiments mimicking real-world conditions. So we should look into more pesticide mixtures based on measured exposures. And then also there are kidney tissue features observed in humans and rats that are likely indicative of nephrotoxicity. And for that, we are currently investigating whether they can be considered as new indicators of nephrotoxicity. We should also maximize collaborative use of samples because many animal studies are being performed. And it would be to the benefit of the community if you could really share samples to allow additional analysis, also in depth towards potential new indicators. And finally, we might consider discussing whether it makes sense to perform mixture studies before marketing. Is this feasible or not? These are some things to look into. So with that, I would like to thank you for your attention. And if there will be any questions, I would be happy to address them.
Thank you very much, Benjamin, for your presentation. I see someone clapping. That's always nice. Thank you. The moment there are no questions in the chat, there is someone that is raising her hand, but that was already a while ago. So, Honor, did you have a question for Benjamin, or was it an old hand?
An old, I'm sorry, an old.
Okay. Then, if there are no questions for Benjamin, we continue and try to be on the schedule. So the next speaker is Nina, and Nina is talking about something that we all are, namely consumers. So Nina, the floor is yours.
Yes, thank you very much. Can you hear me? Okay.
Yes, we can hear you.
So, yes, I'm Nina. I'm a PhD student at the Department of Environmental Science at the Radboud University in the Netherlands. And Benjamin already just briefly mentioned it, but I will be elaborating on the duplicate food portion study, which studied consumer exposure to pesticide residues from food. So pesticides end up in food systems through agricultural use, and via this route also end up on our plates. The average consumer has a complex diet. With this, we mean that it consists of processed and unprocessed products, and also local and imported products. The European Union, and especially the European Food Safety Authority, make sure that there is a safe consumption of pesticides by setting a maximum residue level on food products meaning that there is a maximum amount of pesticide that is allowed to be on these products. From the sprint biomonitoring study that Violette introduced in the beginning of this session, we know that there is a low internal concentration of pesticides in the non-occupationally exposed population. So this is in the people that do not work with pesticides. And with low, we mean that there is a concentration of the pesticides lower than 1 nanogram per milliliter in the blood plasma. Then there's also the case of aggregate exposure and mixture exposure, meaning that consumers are generally exposed to the same pesticide from different food items and exposed to different pesticides at the same time, which is the mixture exposure. To study this better, we have a new method called the duplicate portion analysis, which we collect an exact duplicate of the consumed food alongside urine to study the relationship between pesticide intake and pesticide excretion. And I will elaborate a bit more how this exactly worked. In the duplicate portion analysis study, we asked participants to collect an exact duplicate of their food. So we asked them to prepare the same food twice and place it all into a bucket for 24 hours and also include the beverages, so juice, but also water. And we also asked them to add processed food. So with processed, we mean food that's prepared by the participant by peeling or boiling or grilling. In addition to this, we also asked them to collect all their urine for 24 hours and 12 hours of the next day. We asked them to keep a food diary so we know exactly what is in the bucket of food. We extracted all the pesticides from these food portions and from the urine and analyzed them for the occurrence of pesticides and metabolites. And metabolites are the breakdown products of pesticides after they have passed through the body and went through the liver and kidney. In total, we screened for 203 pesticides, which we also sometimes call parent compounds, in the food portion, and 46 of their metabolites were screened in the urine samples. We did this study in 7 countries, of which 6 in the EU and then Argentina outside of the EU. We looked at consumers, but also neighbors and farmers, since they are technically also consumers. However, the farmer and the neighbor live or work occupationally with pesticides. So they have a higher potential for non-dietary exposure, for example, via inhalation. And this can happen when the farmer, for example, sprays and it ends up at someone living very close to the field, who can also inhale it or maybe touch. And via the skin, this pesticide can then also be absorbed. In total, we recruited 43 participants who all finished the protocol of this study. This figure shows the results of the number of different pesticides that we measured per 24-hour food portion. And what's important to look at in this figure is the dark line in the middle of the box, because it shows the median pesticides. And it's also important to look at the range, which is the difference between the maximum amount that we found and the minimum. And as you can see, for some countries, this range is quite big, and that there's also a difference between countries in the median amount of pesticides that was found in their food. Comparing this to the results of the urine, you can see that this variation is less big, but we also in general found less metabolites per sample. Moving on to the top 10 most often detected pesticides. In the diet, we found mostly fungicides. This is all the names in purple. And this is pesticides that target fungi. And we also found 2 pesticides that work primarily against insects. And number 2 is a co-formulant of an anti-insect pesticide. When we compare this with the urine, we find in urine mostly the insecticides, 1 of the fungicides, and then some herbicides which work against weeds. If you want to link these, you can look at some of the pesticides that end up in the food portion, but also as a metabolite in the urine. For example, this one is metabolized into these 4, which we also nicely measured in our study. And this tebuconazole, number 4, also ends up as a metabolite. To elaborate a bit further on this match and why it's important to study this, I will give the example of this tebuconazole. In 53% of all the participants, we detected the tebuconazole in the diet and also in the urine, which is a very clear relationship between the diet and the urine. and indicates that the dietary exposure is a very reliable route of exposure. However, there are also several pesticides that have no match. For example, we found some pesticides only in urine and not in the diet, and this is a clear indication that there are other exposure routes in play. How can you use data from these duplicate portion studies? If you want to estimate the amount of pesticide that someone takes in the daily, you can analyze the amount of pesticide in the food portion, multiply this by the weight of the food portion, and divide this by the body weight of the participant. And if you have this estimated daily intake, you can compare this with the accepted daily intake, which is the level at which no effects are expected if you are exposed for this amount. In current practice, the EU member states perform regular monitoring of bought food products. And with this, they mean products as you buy them in the store as a whole. Check if these exceed the maximum residue levels, and this leads to figures such as the following. So here you can see for every member state the percentage of non-compliant samples, meaning that how much of their analyzed samples exceed this maximum level. Every year, this is reported in the European Union report, which is publicly accessible. However, there are some differences between the product that you buy and the product that you consume in a real-life scenario. There's food processing in play. As I already explained, this can be peeling, boiling, grilling, or heating the food in any other way, but also portioning. For example, if you buy a bag of apples, you don't consume all apples in one go, but you portion this. Then there's also the case of aggregate exposure. As a consumer, you can be exposed to multiple pesticides to the same pesticide, actually, from multiple items in your food basket. If you want to calculate the ADI exceedance, currently EFSA looks at the concentration of pesticides in the bought food products, sees if they exceed the MRL, and they also apply food processing factors, which make an estimation of the food processing techniques on the amount of pesticides. In the DPA approach, the concentrations of pesticides are measured in the actually consumed food and in the beverages. Food processing is also included within the measured data. Combining the analysis of urine with the DPA approach can also give a good assessment of internal exposure, and in the DPA approach, other exposure routes other than food are also taken in account, such as inhalation or skin, as I explained earlier. Some key messages. The duplicate portion studies combined with human biomonitoring can provide a better insight in real uptake of pesticides by consumers. Current monitoring is applied to bought food products, but duplicate portion study also includes processing and portioning and other routes and sources of exposure other than food, such as inhalation or skin contact. Exposure to the same pesticide from different food items is also covered in this approach, and in short, a combined approach can be a considerable improvement compared to the current monitoring practices by food safety authorities. Thank you for your attention, and I'm happy to take any questions.
Thank you, Nina. So far, no questions. If people realize they do have questions, they can always ask sprint@ver.nl, and we are happy to forward relevant questions to the speakers of today. We're going to...
There's a question in the question and answer segment. Let me see if I can put it in the chat.
I see the question. Should I read it out loud?
Yeah.
So the question is, what can actually be done to ameliorate pesticide concentration in semi-processed or raw agricultural food, as it is a major problem in developing world like Nigeria, whose main route is dietary or dermal exposure of 2,4-D, organochlorides, and organophosphate pesticides? Okay, thank you. That's a very good question. Very interesting to also see the view of Nigeria on this. I would say the most easy way to ameliorate this concentration is less spraying, but I can understand that this is not always the case. So I would say there are some pesticides. I'm not sure if this is also the case for 2,4-D or organophosphates, which are more sensitive to processing. So I would suggest maybe more processing prior to eating, such as boiling or heating in another way. Often that also helps. Yeah, and I see Violette's also typing. So
I would like to add, avoid that pesticides which are forbidden in Europe are imported to Africa. I think this is one of the most problems, and as well, it is very important, although in Europe, we cannot say that everything is fine, but it would be very, very important as well to implement very strict rules all over the world for the use, and as well, then that will reduce strongly the residues in food. I think that is very important in a worldwide assessment. I think we should go on now for the next.
we go. Maaike is the next one. So we just heard about food, and now we go on to the gut where we process the food. So Maaike, the floor is yours.
Yes, today I will be talking about the effect of pesticides on the gut. For example, on the gut microbiome, but also on gut cells directly. As you heard from Nina, the gut is exposed via dietary intake, but also Nina already explained that there are other ways you can be exposed, for example, via water and also via house dust. In Sprint, we actually found that there is lots of different pesticides in house dust, and the gut cells are exposed to pesticides through a combination of these three components. Of course, this is to a very low dose, but you have to consider that this happens every day again, and this long-term low-dose exposure could still have an impact on your health, for example, via the gut microbiome, or via the gut cells, the cells that come directly into contact with the food and water that you take in. And also there's a very important interplay between the microbes in your gut and your own cells in the gut. And this interplay could also potentially be impacted by pesticides. So first I'll talk a bit more about the gut microbiome, which was already introduced by Eoin. Eoin already explained that pesticides can impact the gut microbiome composition. I'm gonna take a little step back and recap that with the gut microbiome, we mean all bacteria and other microorganisms that are living in the gut, and they actually contribute to human health and animal health. For example, the gut microbiome plays an important regulatory role in the immune system, and also the healthy bacteria in your gut can compete with pathogens. However, the gut microbiome can be disturbed by multiple factors, including chemicals such as pesticides. In a disturbed state, a disturbed gut microbiome has been associated with neurodegenerative disorders, metabolic disease, psychiatric disorders, and also, as Eoin explained, there's a link with behavior. However, research into the effect of pesticides on the gut microbiome is currently not included in approval assessments for pesticides. So in Sprint, we thought it was very important to do a large-scale study. So from all the participants in Sprint, we took fecal samples. So we have approximately 700 samples. Those are from farmers and neighbors from farmers, as explained before, but also from consumers that live in the city further away from the farm, from all 11 different countries that participated in Sprint. And from these fecal samples, we were able to measure the pesticide residues and the metabolites in there, but also we were able to extract the DNA from the bacteria that live in the gut. And via this DNA, we can come to a profile of all species that live in the gut microbiome. And we actually did find that for multiple pesticides, including glyphosate and pyrethroids, so some pesticides that are still used very often, but also pesticides that have been banned, like hexachlorobenzene and DDT, they do have an impact on the gut microbiome. And therefore, we think it's very important that in the future, when we look at pesticides to see if they can be approved for a longer period of time, we also need to take into account the gut microbiome. We also looked at the gut cells, as I said before, and to do this, we used a model made from stem cells. They're grown in the lab and called organoids. And you can think about organoids as mini guts. They have the same cell types as the real gut and the same physical properties. But by using these, we can avoid having to use animal models, which is, of course, a better ethical choice. And we use these systems to test the effect of 5 different pesticides. So for the biologists among us, we did this at a molecular level because we looked at gene expression analysis. And one of the observations we had from this is that pesticides can potentially impact gut barrier functioning. The gut epithelium and the mucus laying on top of the gut epithelium forms an important barrier between the food and everything that passes through your intestine and your bloodstream in your body. It keeps potentially harmful bacteria and toxic molecules in the gut and out of the body. And these epithelial cells are tightly bound together by so-called junctions. And these junctions form a very important component of this barrier functioning. If the barrier function is impacted, of course, some bacteria or maybe toxic compounds could enter your bloodstream, which can lead to a lot of inflammation and problems throughout your whole body, but also in the gut area. And we found in our study that 4 out of the 5 pesticides that we tested did actually decrease the production of these junctions. So they potentially severely impacted this gut barrier functioning. And these compounds were glyphosate, fludeoxonil, piperonal butoxide, and cyprodenyl. So that's 4 out of 5 that we tested, which I think only illustrates that we really need to consider the effect that pesticides have on the gut, which is normally not really assessed. So to recap, the gut microbiome is very important for your health, but as Eoin showed, a lot of literature has already shown that it can be impacted by pesticides, and that's also what we saw within the big SPRINT consortium. So we think the gut microbiome effects should definitely be assessed before approval of pesticides. And we also use a new, relatively new system called organoids, with which we were able to reveal effects of pesticides on gut cells directly without the need for animal experiments. We show that gut health can be impacted by pesticides and therefore should definitely be considered before approving compounds for agricultural application. Thank you.
Thank you so much, Maaike. I'm looking at both chats now, and I don't think there is a question at the moment. There are a lot of people clapping, so they did like your presentation. Thank you so much. Well, we are a little bit behind schedule, so I suggest just to continue with the next speaker. On the original program, it was Daniel that would tell us something. Unfortunately, he got sick today, but luckily we have Paul substituting for Daniel, and he will present his presentation. So thank you very much, Paul, for your willingness to fill in this spot. And I would say the floor is yours.
Thank you very much, Paula. I will talk on behalf of Daniel and explain more about the dietary exposure, the topic that Nina touched upon, but now I will add the contribution of non-dietary external exposure. And I will do that first by showing you some results of measurements. And then secondly, I will also address the calculations in models. And the first say measurements, I would like to just explain that during the sprint project, we have given people these silicone wristbands and asked them to wear these wristbands during 1 week. And these wristbands were then analyzed to assess the contribution of non-dietary exposure. That means exposure by inhalation and by direct skin contact. And we have compared the concentrations of metabolites in urine, so metabolites of pesticides, with the results from the dietary exposure, as already explained by Nina. And we have done additional analyses regarding the contribution of the wristband exposure. For those results in wristbands, we could see that when we found more of these substances that I show you here, 5 different types of pesticides, we saw that the concentrations in urine of these persons were also higher. For another 3 pesticides, we could see that the dietary exposure, when the exposure in the diet was higher, we saw this was reflected in higher metabolite concentrations in urine. So you can see that actually there are different substances that are taken up by different routes of exposure from different sources of exposure. Let's now do some further calculation to make a calculated estimate of the contribution of the dietary exposure and compare that with the different sources from non-dietary exposure, as shown here. Inhalation from the gas phase or from particles, inhalation of or direct contact of dust on skin, and the contact of substances in the gas phase and penetration via skin. So these are all what we call environmental or non-dietary exposure routes. We did these calculations for this selection of pesticides that they were all already studied in the results that Nina showed. And this is a bit busy slide, but trying to summarize what our findings. So for 7 plant protection products, we noticed that more than 90% of the total exposure could be explained by ingestion from food, with only a minor addition from ingestion of dust from an environmental source. For 4 pesticides, as you can see here, also including glyphosate and a degradation product of glyphosate, AMPA, we saw that actually dust ingestion can be a very important route of exposure. And also in this case, the skin contact, direct contact with skin surface was a main contributor. Then for 2 substances that are already phased out in Europe, we also noticed that there was a contribution from gas to skin exposure. And last but not least, we studied a substance cypermethrin and a substance that's not used anymore, hexachlorobenzene, and we found that for these substances there was primary exposure from particles to skin, and to a lesser extent also dust ingestion and inhalation. To try to compare these different contributions in mixtures. So let's say adding all together the contribution of all these sources and routes of exposure, we try to in a let's say worst case, try to compare the total exposure from dietary, non-dietary exposure for a mixture of all these substances. And we found that for this mixture, the risk of exceeding the acceptable daily intake was about 1%. Key messages. I think that I've shown that for some pesticides, we could find an association between the levels of found in urine with the levels that we found in wristbands and diet. And we looked at the maximum levels allowed in food, and also included environmental exposures that are not current part of the current system in European Union. And then we also looked and found that certain pesticides are also taken up by dust ingestion and inhalation that can be, for some pesticides, play a prominent role in that's more important than we initially expected. And last but not least, I think that we think that adding all the pesticide exposure from different sources and routes, that we can think of certain scenarios where the acceptable daily intake could be overstepped. And at the moment, we don't know how often this would occur. Thank you very much for your attention. I'm happy to answer any of your questions.
Thank you very much, Paul, also for substituting again and sticking to the time. Very nice. I don't see a hand raised nor a question in the chat, so I would suggest to go on to the final part of this session. I will share my screen so Violette can have the final discussion.
Yeah, I hope you enjoyed these different approaches we were following in the Sprint project. And please ask us or send a mail to sprint@vuur.nl for any questions. We will then send them to the speakers here, and as well, if you have, if you would like to disseminate, we will make the recording available as well on the Sprint website, so that you can distribute it into your network, and as well, that we are always open to answer questions. Now I come to the next slide and to see, okay, what are the main outcomes, what we would like to communicate with you. Until now, we still have 1 year to go in Sprint, so the final session will be there next year. But until now, what we can say is we compared the Sprint results towards the EFSA regulations, which is a European regulation or authorization for pesticides. Due to the EFSA approach to assess human exposure or risks, we see here a limitation because EFSA in this moment only has defined maximum residue levels in food for single pesticide residues, but as Nina and showed and Paul as well, yeah, and the exposure is much higher, so we are mainly exposed to mixtures, and there is no maximum residue level for mixtures, nor for total number of pesticides, nor for concentrations. As Paul clearly showed in his last presentation, the acceptable daily intake for humans is as well based on single pesticide residues in food and drinking water, but the exposure by air indoor dust is not taken into consideration. And when Sprint, our colleagues from the human health are strongly involved now to improve this possibility and methods to be as well included into the EFSA regulations in future. What as well we have seen is the risk of pesticide mixtures, and there I'm talking even about those who are approved for common use, is not accessed. So the risk, the health risk for each single pesticide is assessed, but farmers often use mixtures. And as we heard from Peter Fantke, a lot of pesticides are used per crop, varying between in sprint, we saw varying and from 7 to 25 or 30 pesticides per crop. That means the farmers are exposed in one growing season of even in one moment when they make the mixtures to these, and this risk is not known. That the EFSA underestimates human exposure, not taking this non-dietary exposure into consideration. The risk related to exposure to the high number of residues is not considered. And the question what we ask ourselves in sprint is, can we assess the real risk for humans, what humans are exposed to? Because as we could show in the houses of one farmers, we find up to 120 different pesticide residues, and that is only in one house. So we will be able somehow to really see what is the risk of the residues we find in the real world on human health. So what about the environment here? For the environment, there are no benchmarks for the number of concentrations of residues in soils, nor in sediment, nor in air or indoor dust, and neither for the concentrations. So if a farmer wants to shift to organic farming, there is no rules how many pesticides can be there in the soils. There is not known how many are uptaken by the plants. So there is no limit for the whole environment, the health for water. And as shown by Nelson and by Kayode, now by EFSA, there is no multi-pesticide risk assessment, even not for pesticides as mentioned, that are applied as mixtures and directly coming together to the soils or entering directly together into the aquatic environment. And as Nelson and Kayode very clearly showed, is that the EFSA species, which are used for risk assessment, and the EFSA indicators are not as sensitive as native ones or more sensitive indicators, as could be nicely shown for the zebrafish. So our recommendations until now: if we apply the precautionary principle to protect ecosystem and human health, this would mean that we have drastically to reduce the use of pesticides. Regulations should be adapted towards risk assessment based on real-world data, taking into consideration all sources and exposure pathways related to mixtures. And what as well is very important, what Peter Fantke showed in other presentations already, that it is very important to include health costs derived from pesticide applications into cost-benefit analysis. Sheep food seems to be sheep food, but if we consider the real trade-offs, for example, human health effects or environmental health effects, and sum them up, then we see that cheap reproduced food is not surely the cheapest one. And last but not least, there are concepts for a modern agriculture 2030, including agroecological systems, connecting them with robotics and precision farmings. These concepts exist, and policy is urgently asked to implement them. As you can see, if there are questions, discussions, ideas, please contact us. And I don't know whether there are questions or comments to my recommendations now. If I see one in the chat, but I don't know. No, this is from Jane. So if not, I would like really to thank you a lot, all of you, to having attended this meeting. Please disseminate the recordings you will be getting by Paula. Paula will take care of this. These recordings will be disseminated. Send these around to your colleagues to inform them that this meeting has taken place and that we are open for discussions and we like to get input from your side. Thanks a lot again. And yeah, Paula, I don't know whether you would like to say some final words.
Well, no, actually, just thanks to the speakers that gave a lot of input. Thanks for the people that joined us in this session. And as told, I will probably make a separate website item on the Sprint website. So, but you will hear more about it.
Okay, have a nice evening or morning, wherever you are. Thanks a lot and hope to see you again next year. Bye.
Bye.